Thank you all for coming. Thank you. Thank you, Keith, for this amazing introduction. And I feel like my face getting red. And thank you, Susan, for the opportunity invite me here and for the opportunity to present here. And thank you, Larry, for all these journeys to study herpes. So first, I have no disclosures. And today my talk is all about genital herpes. So I will share with you 3 stories. The first is about the mapping the spatiotemporal dynamics of the battlefield in human skin that infect with herpes simplex virus. And the second, I will present you a bioengineered vascularized skin on chip model for in vitro modeling of HSV infection. And the third story is the most recent collaboration with NCATS on using 3D bioprinted human skin equivalent for antiviral drug discovery. So genital herpes is highly prevalent sexually transmitted disease that caused by herpes simplex virus infection. And HSV is a double-strand DNA virus that encodes more than 80 proteins, and there's 2 subtypes, HSV1 and HSV2. Although they share more than 80% sequence homology, they differ in serology, clinical manifestation, and the transmission route. So while HSV1 is known for causing cold sore in oral mucosal and keratitis in eyes, HSV2 is credited for sexually transmitted genital herpes, but both HSV1 and HSV2 can cause genital herpes. So this is actually which has a very substantial global economic burden and also have worldwide public health impact. As of 2020, 2/3 of the global population are infected with HSV1, and 10% of them have HSV1 genital herpes. And there's more than 520 million people are living with HSV2 genital herpes. This translates into 1 in every 5 people aged between 14 and 49 have genital herpes infection. So both types can cause neonatal herpes that has very severe neurological consequences. And HSV2 is a significant risk factor for HIV acquisition and the transmission. In terms of treatment, right now we have antiviral therapeutics like acyclovir, but there's increased antiviral drug resistance occurring in the immune-compromised patient. So far, there's no vaccine available, and all the candidate vaccines that show great promise in the murine model, they all failed to protect in human clinical trials. So that is really emphasizing the importance to understand HSV pathogenesis in humans. So we know that HSV first enters the human body through a breach in the skin and the mucosal surface. So it replicates in the epithelial cells, and then the viral progeny gains access to the innervating nerve network and reaching the nucleus of sensory neuron will establish lifelong latent infection. But under conditions like stress, sunburn, and the neurological injury, HSV can reactivate from this latent state and be transported back to the peripheral, where viral shedding can occur in the form of ulcerative lesion or completely asymptomatic. So both forms of reactivation can lead to viral transmission to their sexual partner and also vertically from mother to infant. So what are the characteristics of human HSV2 reactivation in time and space? So the study led by our own doctors, Dr. Anna Wall, Karen Mark, and Christine Johnson, have shown that HSV reactivation is frequent, and they occurs mostly asymptomatic and is short-lasting, and half of those shedding episodes last less than 12 hours. And the viral shedding distribution and the clinical outcome can also vary widely, from completely asymptomatic to sporadic short-lasting recurrence, and to frequent and severe ulcerations that require suppressive therapy. But above all, the asymptomatic shedding is the most common form of HSV reactivation in humans. So my research focused on understanding the local immune battlefields, which we believe could influence the frequency and the disease severity of HSV2 reactivation. So we aim to understand the immune mechanism that underlying this rapid clearance and viral resolution in asymptomatic shedding versus those that cause tissue damage, and the immune pathology in also forming herpes lesion. So in this standoff between the virus and the massive infiltrating immune cells, we want to understand whether the immune composition, the difference in immune composition, spatial distribution, and effect function can lead to distinct disease outcome, whether it's asymptomatic or also forming herpes lesions. So first, I will summarize our published work on T-cell mediated protective immunity during human HSV2 infection. So we'll first discuss the tissue-resident memory T cells and their role in controlling HSV infection. So my first project, when recruited by Larry, was to do in situ tetramer staining to detect the presence and distribution of HSV-specific CD8 T cells. But instead of doing tetramer staining, we developed quantum dots nanoparticle conjugated peptide MHC multimers to overcome this high fluorescence background in human skin, and also to increase the photostability during this prolonged confocal imaging. So we detect HSV-specific CD8 T cells throughout the during healed lesions in the epidermis, dermal-epidermal junction, and in the dermis. And what's interesting is even weeks after lesion completely healed, we see the CD8 T cells persist in local tissue, and including the antigen-specific, HSV2 antigen-specific CD8 T cells. And those T cells, they localize to the dermal-epidermal junction, and they have close interactions with the basal keratinocytes next to the free nerve endings, and they are also in close contact with terminal Schwann cells in the nerve branches. So this, because we know viral particles, reactivating viral particles are released into through the sensory nerve ending into the peripheral. So we hypothesize that the CD8 T cells, by position themselves between the reactivating virus and the target cells, is to optimally control the viral infection. But then we also further examine this T cell persistence in individuals during a severe antiviral therapy. We found both CD4 and the CD8 T cells persist in tissues for more than 20 weeks. That's how far we have examined. And that CD8 T cells lining up the dermal-epidermal junction, while the CD4 T cells are in the epidermis and near blood vessels. And we realized this persisting CD4 and CD8 T cells, they form immune clusters with dendritic cells including the plasmacytoid dendritic cells and the DC sign positive dendritic cells. So this T cell DC close interaction in local tissue is not affected by acyclovir treatment. So while this interaction might be important for the long-term persistence and the function of those local T cells, it also provides perfect target for HIV infection. So at that time, there's 2 large international clinical trials, including the one that conducted at the University of Washington by Dr. Connie Callan and Anna Wall and Dr. Larry Corey, and they showed that acyclovir suppressive therapy failed to reduce HIV incidence. And our study showed that acyclovir treatment does not alter the local immune milieus in terms of their persistence, distribution, activation, and interaction. So the persistence of this HIV receptor positive cells could be the potential mechanism for increased HIV acquisition by HSV infection. So we then, to determine the in vivo, what's the function of those CD8 cells at the dermal-epidermal junction. To do that, we developed a cell type specific laser capture microdissection. So Kay and Tao teamed up to laser microdissect individual CD8 T cells from this dermal-epidermal junction and compared their transcription profiles to CD8 T cells that isolate from control tissues. And we show that DEJ CD8 T cells expressed high level of signature genes in T cell activation, cytolytic function, and cytokine and chemokine expression. So indeed, the upregulation of interferon gamma, TNF alpha, RANTES, along with perforin and granzymes, really indicate those DEJ CD8 T cells are effector T cells. They constantly engaging and eliminating viral infected cells. So in asymptomatic HSV reactivation, the viral DNA can be readily detected by PCR in normal appearing skin tissue. And we can also detect limited viral protein expression can occur in multiple keratinocytes in the epidermis. And we see that CD8 T cells and HSV infected cells form clusters. And there's a high CD8 effect to target ratios and elevated cytolytic granule expression were observed. And we also show that CD8 alpha T cells can also accumulate in this space, and the CD8 alpha beta T cells are getting recruited to the dermal-epidermal junction. So this is in great contrast with the also forming herpes lesion, where very few CD8 T cells were detected in the infected area. So this data indicate that DEJ CD8 T cells, they are actively perform immune surveillance and rapid containment of the virus. We then also use T cell receptor sequencing and the TCR repertoire analysis to demonstrate the tissue resident nature of this DEJ CD8 T cells. So among these 6 biopsies collected from the same individual over a 2.5-year period, they have 4 different shedding episodes, and from different locations, whether it's labia, perineal, or buttock, and also at a different stage of reactivation and healing, from lesions to all the way to 8-week post-healing. So what we found is we detect shared CDR3 sequence in multiple biopsies over this 2.5-year period. And there are T-cell clones that are present in the skin at all time and through all the tissue locations. So this really demonstrates that those DEJ CD8 T cells are tissue resident in the local tissues. So this human study provide essential evidence that the tissue resident memory T cells function as the first responders to recurrent HSV infection in the peripheral tissue. And they also support the field in establishing this concept of tissue resident memory at its early stage. Then we ask, how do TRMs influence the skin microenvironment and therefore provide tissue-wide protection? So to do that, again, we use laser capture microdissection, but this time is we isolate individual basal keratinocytes that is surrounding the CD8 T cells from post-healed tissue. And we compared their gene expression profile to keratinocytes that isolate from control skin. So there's more than 1,200 genes were differentially expressed. And genes involved in viral infection in the infectious disease category was the most significantly enriched in the functional annotation analysis. So the genes involved in host gene transcription, their RNA transportation and processing are all downregulated. But genes involved in interferon defense and also the Cas-based activity and epigenetic repression are upregulated. So from this data, we think that CD8 T cells, they actively communicate with the surrounding neighboring keratinocytes. We then identified an antiviral gene signature in keratinocytes that when exposed to interferon gamma treatment. So these signature genes are involved in antigen presentation, chemotaxis, and also antiviral restriction. Importantly, that viral infection itself does not activate those signature gene expression. So, and we also find that HSV infection was significantly delayed and reduced, GFP expressions reduced when cell was pre-treated with interferon gamma and compared to those untreated keratinocytes. Importantly, this interferon gamma signature gene expression can be readily detected, observed in human skin. And we see this elevated level and increased breadth of this signature genes in 8-week post-healed tissues when the interferon gamma can be detected in the DEJ CD8 T cells. And overall, it's about 2-fold increase in the signature gene expression level. And among them, there's a gene called IFI16. So IFI16 is an interferon gamma inducible protein, and it is known for sensing viral infection and restricting HSV gene expression. So we see that IFI16 protein was widely expressed in this histologically normal appearing epidermis that distal to the ulcer-bearing regions. And even weeks after lesion resolution, and we see heightened IFI16 expression in those post-healed tissue, but rare in normal controls. So this data support that CD8 TRMs could, through interferon gamma expression, to provide widespread protection in surrounding skin epidermis. So, and we also test that in a larger cohort of 22 subjects, this collective level and the hierarchical pattern of this, cell intrinsic antiviral signature is extremely similar across all the controls. But in tissues, in post-healed tissues, we see elevated expression of this signature genes. And this associated very well with increased DEJ CD8 numbers and their interferon gamma expression. So we think this cell intrinsic antiviral signature could serve as a surrogate biomarker for immune protection at the skin barrier surface. So to summarize this first part of the talk, that I have shown you that HSV specific CD8 T cells, they persist in tissues after herpes lesion healed. And there's a high CD8 effect to target ratios, and also increased cytotoxicity are evident in the asymptomatic shedding. So, and we see that DEJ CD8 T cells, through interferon gamma production, can instruct the enabling keratinocytes to induce cell intrinsic immunity. This including many antiviral restriction factors to resist viral infection and then prevent viral spreading and achieve tissue-wide protection. When in areas of skin, when lack of those TRM CD8 T cells, the HSV can quickly disarm type 1 interferon response and to achieve productive infection and the rapid viral growth, result the viral manifestation and the lesion formation. So now I'm going to switch directions and discuss a 3D vascularized skin-on-chip platform for modeling HSV infection in vitro. So this work is spearheaded by a talented postdoc, Jia Zhu, who has a PhD training in the Department of Bioengineering at UW. So I was very fortunate to have her. So to bioengineer a 3D skin, human skin mimetic, so we aim to recapitulate natural skin architecture and function. So this including to have a stratified epidermis on top, and in the dermis to have collagen and the fibroblast for structure and mechanical support. And also we include a vascularized vasculature for circulation. So a desired engineered skin should retain the barrier function and also be responsive to infection and to permeable for drug and immune cell infiltration. So, and it should also allow us to monitor the viral infection and immune response in real time and to serve as a preclinical platform for assessing drug efficacy and other therapeutic agents. So this is how the device looked like in real time and in fluorescent confocal image. So H&E staining indicates that our engineered cells has a typical stratified epidermis that including the basal layer, the spinous layer, granular layer, and the conified layer. So, and we also use immune fluorescence staining to confirm the key markers for key marker expressions in our skin on chip and compare them to the natural skin. And we used CK10 for differentiated keratinocytes, CK14 for basal keratinocytes. We can also detect basement formation, and also we see the vermetin expression in the fibroblast cells. So we then infect the skin chip with a GFP expression herpes. So inoculating of HSV direct on top of the skin results no infection. So this indicates our skin has a barrier function of this, our stratified epidermis has a barrier function. So we have to use a mechanical punch to create a physical disruption to allow virus to have access to the susceptible cells. And we found that infection mainly occur near the tissue ruptures, and we see viral spread. We also see viral spread mainly occur in the basal layer of the keratinocytes. The keratinocytes that express CK14 but not CK10. And the confocal image also show that HSV infection in the skin chip induce morphological changes, such as the formation of those multinucleation and also the enlargement of the nuclei and the chromatin margination. So this is very similar to what we see in the herpes lesion. So we also want to evaluate the role of keratinocytes differentiation in HSV infectivity. So then we infect epitheliums with HSV at a different stage of keratinocytes differentiation. So the GFP expression indicates that undifferentiated keratinocytes is the most susceptible for HSV infection. Even 1 day after air-liquid interface, we see dramatic reduction. There's 70% reduction in GFP expression. And after 7 days of differentiation, there's almost none keratinocytes were found to be infected. So this result really indicate that basal keratinocytes is the most susceptible target for HSV. And those non-replicating differentiated cells, they are more resistant to HSV infection. So this data also highlight the importance of the intact barrier function in preventing viral infection. So next, we examined the antiviral activity of acyclovir. So we perfused acyclovir through the vascular network at different concentration, and we demonstrate acyclovir has a dose-dependent antiviral inhibition, and we also test the timing of acyclovir treatment. So here you can see perfusion of acyclovir 24 hours after the initial HSV infection, which is usually the case as when we treat with herpes, right, after the outbreak. But it is less effective than perfusing acyclovir at the time of infection, or even like 24 hours before the viral infection. So you do want to treat herpes early. The moment you get the tingling or the symptoms, you want to use acyclovir as early as possible. So we think this platform can be used for preclinical efficacy testing. And we also validate whether the endothelialized, this blood vessel network is competent for mediating immune response. So we perfuse freshly isolated neutrophil through the microvascular network 1 hour after viral infection. So we monitor the migration of neutrophil and their extravasation by real-time imaging. So we actually can see even at the 4-hour post-infection, we already see plenty of neutrophils migrate out of the vessel and enter into the dermal space. And at the 20-hour post-infection, majority of the neutrophils already extravasated, and they migrate upwards against gravity to really trying to engulf viral infected cells. And we detect 3 cytokines. And among these 3 cytokines, IL-8 is the most abundant detected. So when we perfuse antibody neutralizing the IL-8, neutralizing antibody, we see we can completely abolish the neutrophil migration and extravasation and their attachment. So even the neutrophil, the cellular skeleton structure has changed from this elongated active form to a rounded resting form. So this experiment indicates that neutrophil reaction to HSV infection is IL-8 dependent. So to summarize this part of talk, so we engineered 3D microfluidic device that has mimic the human skin architecture with epidermis, stratified epidermis, and dermis with a microvessel network. And the network can form a functional lumens with integrity and permeability, and can be used for perfusion. And we see that viral infection of this system has exhibit characteristics of viral infection that we see in human genital herpes. And this device is responsive for acyclovir treatment and also competent for immune cell transendothelial migration. So lastly, I'll present you our recent collaboration with NCATS, with the Mark Ferris group, to use the 3D bioprinted human skin equivalents for antiviral drug discovery. So acyclovir is a nucleoside analog of guanosine. So acyclovir needs herpes semitic kinase to first to convert to phosphorylate into a monophosphate. And after that, there's enzymes, host enzyme is responsible for adding 2 more phosphate to make into a triphosphate. And then they can compete with triphosphate, guanosine triphosphate, and then to be incorporated into the viral genome and then stopped viral replication. So, so acyclovir can, reduce the duration and shorten and reduce the severity, and it is also it has. But acyclovir has limited efficacy in preventing subclinical HSV reactivation. So it is also, as I mentioned before, they fail to increase the risk of HIV acquisition and transmission. And they face drug resistance in immune-compromised patients, particularly in those that undergoing allogenic hematopoietic stem cell transplantation. So we need new antivirals. So, but then conventional antiviral drug discovery use viral cells and the fibroblast cells. But we have shown that HSV, they manifest in human skin, and through our in vivo biopsy study and our in vitro skin on chip study. So the skin keratinocytes is the primary target that encountered by the virus, and also the most susceptible target for viral infection. So then we decide to isolate keratinocytes and the fibroblast cells from these 3 millimeter punch biopsies, and then infect them. And we did from 6 adult donors, and to investigate whether acyclovir display similar efficacy in keratinocytes, fibroblast cells, and the viral cells. So Ian, postdoc in the lab, he found that HSV infection and the GFP expression are more rapidly occur in keratinocytes, then they're donor matching fibroblast or viral cells. So the GFP peak at the 20-hour in keratinocytes, 36-hour in viral cells, and 48 hours in fibroblast cells. And also the time to initiate the GFP expression is significantly earlier in keratinocytes. So it's 6 hours. 6 hour in keratinocytes, 7.9 hour in fibroblast cells. And so also GFP increase much faster in keratinocytes than fibroblast cells and the viral cells. So he then evaluate acyclovir antiviral potency and efficacy using this donor-derived keratinocytes, fibroblast cells, and compared to viral cells. So the dose response curve indicate that acyclovir is significantly less potent in keratinocytes than fibroblast cells and viral cells. The concentration, the IC50 for keratinocytes is about 200-fold higher than what is needed for fibroblast cells and the viral cell. 60-fold for the compared to the viral cells. So that is really telling us that we need a physiologically relevant system to screen antiviral drug screening. So in order to create this physiological relevant in vitro human skin model for drug discovery, so we collaborate with Mark and his group at NCATS, and Corey is the first author of our recent impressed paper, using this 3D bioprinted human skin equivalents in a 96-well platform for HSV infection and antiviral screening. So we have two infection models. We use two different infection models, the submerged model and also we call the alley model. So the submerged model is we have a single layer of basal keratinocytes. So the infection is through apical infection, which we want to mimic the primary infection. And the alley model, we infect through basal lateral route. We want to, and in the alley model, it has a stratified epidermis on top. So we want to model that for viral reactivation. So in reality, we found that submerged model that only have keratinocytes get infected, while the alley model will have only the fibroblast cells get infected. So we use both model system in our screening. And this is our layout of our 96-well plates. And we including both positive control and the negative control on each plate to account for any variations in screening. So we implement a primary screen of 738 compounds with a single dose of 10 micromolar using 3,840 bioprinted human skin equivalents. And from that initial screening, we identified 106 hits and then performed a secondary screening. In the secondary screening, we use a dose response from 10 micromolar all the way to 0.04 micromolar. So, and then each compound were characterized by this concentration response curve, we call the CRC class, according to their potency and efficacy. So this led us to select further 41 candidate compounds. And of the 41 compounds, 23 of them already been reported or published. So this including 5 all the 5 cyclovirs and the 2 helicase primase inhibitor, which is the amidomavir and also the pritilavir. So amidomavir already being approved in Japan early this year for treating HSV infection, and pritilavir is still in phase 3 trials in the U.S. for immune-compromised patients. So, and then we select top 11 candidates to further our pharmacological, to establish their pharmacological profiles. So these are the 11 candidates. So, so then Ian investigate this top candidate and compare their antiviral activity to acyclovir. And all these 11 candidate antivirals are more potent than acyclovir in keratinocytes. But the 5 of them are more potent even than acyclovir in fibroblast cells as well. So, so from this we know that, and these 8 compounds, they show significant difference between keratinocytes and fibroblast cells in terms of their antiviral potency. So, and we found the 2 compounds actually they fail to inhibit HSV in fibroblast cells. So really the cell types and it really matters. So Yui, a statistician, he helped us to perform systematic comparison between all 4 models, the 2D keratinocytes, 2D fibroblast cells, the 3D submerged and 3D alley model, to compare the potency, toxicity, and the selectivity. So what is shown here, that the impact of the cell type and the 2D, 3D model on the antiviral potency are compound specific. So we see that acyclovir was most significantly affected by cell types and also 2D, 3D environment, while the amidomavir, pritilavir, they have exhibit less than 3-fold difference among all the model tested. So, and we also find the keratinocytes 2D model is most susceptible for cytotoxicity, and the 3D model is more resistant to cytotoxicity. So, so this data really indicates that cell type matters and 2D, 3D model system matters. So lastly, as a proof of concept, we bioprinted a donor-derived keratinocytes to generate donor-specific human skin equivalents. So the donor-derived keratinocytes successfully develop into stratified epidermis in this adult alley model, and they have CK10 and CK14 expression, and all these 11 candidates, they show similar potency in antiviral, in HSV antiviral activity. So this allows us to establish the strategy that we can use neonatal cells for large screening, while at the early phase of the drug discovery, and later on we can incorporate adult human-derived keratinocytes and fibroblasts for further testing and preclinical evaluation. So to summarize this part of work, so HSV, we show that HSV replicates significantly faster in the primary human keratinocytes than donor-matching fibroblast cells and viral cells. And acyclovir is less potent in keratinocytes, which is the most important cell type for HSV infection. And the helicase primase inhibitor are potent in both 2D and 3D models and with little effect or variation in cell types. So we think the host targeting antiviral candidate should be evaluated using physiological relevant system, and we are very excited that adult donor cells can be incorporated into the 3D bioprinting, and so allow us to counting for the variable genetic background in drug discovery and early in the drug discovery. So lastly, I want to thank all the people that contribute to this work. And CJ is the one that spearhead with the skin on chip work, and EN, and now the Angelina is also working on the keratinocytes. And we have Wanyi and Joe joined the lab to do more advanced skin on chip and also the 10X work. And we have Kay and Tao did a lot of early work out the TRM characterization. And we still have a lot of exciting works and for using more advanced technology, and I'm sure I will show you next time when there's opportunity. And thank you all for coming, and we have some business for evaluation. So thank you. Okay. Any questions? Hi, Susan. Yeah.
That was a wonderful talk. Thank you so much. So you showed that the CD8, this is relates to the first part of your talk, the CD8 tissue-resident memory cells correlate with reactivation when there's a high CD8 to target ratio. And my question is, do you have a sense as to why some individuals have a lot of those tissue-resident memory cells and others don't? And is it proportional to the CD8s in circulation?
Yeah. So right now we're doing a study. Like really, we have a cohort of 38 patients. They have very different shedding rate. Some from only 5% shedding, while the highest has like 75% of days they shed. So we are studying. We are using multiplexing imaging to analyze different immune cells, the CD8 T cells, CD4, and also neutrophil, macrophage, monocytes, and then see whether there's any correlations. And we actually found there is some correlation in terms of the number of both CD4 and the CD8 T cells as they in the lesions, to show how close to the epidermis. So I think the ability of, maybe this is also related to keratinocytes, the ability of keratinocytes to call in the T cells really matters, because it's kind of like a race between the virus and the host. If there's slight difference in recruiting the immune cells in, and I think it will make a big difference. Yeah. But great question. Thank you. Okay, Cindy.
So for the T cells, are there certain peptides that are good to present that maybe are associated with control or better outcomes, or certain agility times, something that you can associate with that?
Yeah, that's a great question, right? So initially when we do the TCR, so the TCR repertoire, so we see there's a lot of different oligoclonal. So the T cell has oligoclonal. So that means they might recognize different peptides. So right now we have another junior PI, Emily Ford, who in collaboration with David Cole, they're trying to tease out what are the peptides those T cell recognize. Yeah.
Oh, that seems like that would be really interesting because, it could be that broad recognition is good, or it could be that there's some focus on certain regions or certain viral proteins. I think that matter. So it'd be really interesting.
Yeah. But I think currently from those people, from people who has very frequent reactivation, they have very strong oligoclonal profile. So maybe you need a diversity. The more diversified, the repertoire will be better. But we'll see. Yeah. Hey, Keith.
So I've always been fascinated with the finding. You really are just some brief this surveillance of T cells right at the tips of the intervening axons.
Yeah.
And this is in a recurrence where the virus is reintroduced into the susceptible site.
Yeah.
I was curious as to whether neurons could actually be introduced into some of your skin and chip or similar sorts of models to try to
I know.
investigate that.
Yeah, and I think that's the future direction. So right now we have this skin on chip. I think I want to introduce like innovated skin on chip. So if we can introduce neurons, and particularly now people can differentiate from fibroblast cells to those, can even directly differentiate from fibroblasts to sensory neurons. So if we can do that, then we can have autologous system and to study immune response. I think I'm going to write that grant.
We have first, by the way, over 50 people participating by Zoom. So,
Oh, thank you.
A large group of people who are not here. One of whom has a question. So Steve Poliak asked, very nice talk. Do you predict that you'll see similar differences in cellular susceptibility to other viruses that infect this human?
Okay, I think he's asking for alpha virus. I think it's a good idea to test in physiological relevant cell types. Yeah. And I think because as I read for all those drug development, right? So it costs now $1 billion for develop one drug that can be out in the market. So I think if we can do during the early development, if we can incorporate of this kind of work, and I think it probably will shorten the time, save cost. I think probably this is also why now FDA want to really promote like those organ on chip or organoid system. Yeah, great question, Steve. Hi, Larry.
I would obviously do full cost. Have you ever put zoster virus in your skin, Chairman? And isn't much different.
Yeah, I have not, but that is a great idea. One thing is I don't know, because for herpes, the replication is fast. 48 hours, we can see all the infections. And for the zoster virus, they probably infect fibroblast cells a lot, right? It will be great to maybe talk to David and see. But I think even for zoster infection, we don't know a lot, like in terms of the immune response, acute or later. I think this is technical challenge to biopsy people, who's already in such a pain, right, during their
Yes.
but
I say it because I'm often asked the following is that for you guys are getting more complicated in how herpes works. There are all sorts of cells and all sorts of things going on. Yeah, with zoster, we took a single printing gene, gave it in a labyrinth, and injected in people's eye every single one.
yeah, and worked. Yeah.
Are you overthinking this?
Yeah. I was thinking, I do think about that. Why zoster vaccine is so successful, but then herpes is so challenging, right? So I was thinking first, it could be herpes infection, you have the nerves directly, like a DoorDash, directly deliver the food into keratinocytes, And for zoster, the infection period are much slower, I think, compared to herpes. So it takes time, and then that allows the host to also migrate in. And also maybe there's some difference, could be like between innate versus adaptive. So maybe neutrophils, that what we see during herpes infection, can also contribute to, immune-mediated tissue destruction. But I don't know whether zosters, it's more like T-cell mediated versus innate. Yeah. So, but those are great questions. You should write another grant for that. Okay. Oh, well, thank you so much.